Nucleic acid bonding. DNA is formed from two strands of nucleotides. The two strands are held together by hydrogen bonds between complementary base pairs. While RNA strands fold in on themselves to form unique shapes, DNA is made of two complementary strands which form a double-stranded helix. Complementary bonding in DNA forms between guanine and cytosine and between adenine and thymine. The double strand forms a double helix due to the bond angles in the sugar phosphate backbone. DNA strands will only bond with other strands that have complementary sequences, guanine and cytosine, and adenine to thymine. For example, GTCA must bond with CAGT on the opposite strand. Nucleotide chains are oriented in opposite directions. They're anti-parallel. Each strand goes from its 3' end to its 5' end. At the ends of a DNA molecule, the 5'C of deoxyribose on one strand is opposite the 3'C of deoxyribose on the other strand. The covalent bonds of the backbone and hydrogen bonds of complementary strands are key to the operation of DNA. The strong covalent bonds in the sugar phosphate backbone create a stable molecule, ideal for long-term storage. The weaker hydrogen bonds allow the DNA molecule to be split, copied, and then put together. For both DNA and RNA, the sugar phosphate backbone, shown as gray ribbons here, stabilizes the molecule. Each strand is unique due to its sequence of bases. In this way, genetic information is stored in the sequence of nucleotides. Since the bases are not part of the sugar or the phosphodiester bond, the base sequence is independent of the backbone. Any base sequence is possible. RNA strands fold in on themselves, forming hydrogen bonds between nitrogenous bases and between the nucleotides and the surrounding water. These bonds cause RNA molecules to form different shapes. Different sequences of bases leads to different folding and to different shapes, which leads to different functions. Folding results from the formation of hydrogen bonds between bases that have complementary structures. An adenine base will hydrogen bond with a uracil base. A guanine base will bond with a cytosine base. The bases of DNA are protected inside the double helix, but its helical structure means that DNA's information is less accessible. DNA is a library of code, but the only way that its information can be used is via RNA. RNA is less stable than DNA, but more chemically active. DNA is useful and stable as an archive. RNA is useful working in the cell, yet has a short half-life. DNA is maintained in a safe environment to maintain the integrity of the genetic code. RNA is used throughout the cell to implement the genetic code that is stored within DNA. Compare and contrast the structures and functions of RNA and DNA. What are the elements found in RNA and DNA? State two differences between the backbones of RNA and DNA. How is their complementary base pairing different and how is it the same? How are their shapes different? How do they differ in stability? How are their locations in the cell different? The elements found in RNA and DNA are carbon, oxygen, hydrogen, nitrogen, and phosphorus. Two differences between the backbones of RNA and DNA. RNA uses ribose, while DNA uses deoxyribose. RNA has one backbone, while DNA has two backbones. How is their complementary base pairing different and how is it the same? RNA uses AU, while DNA uses AT. Both use CG. Both use hydrogen bonds to form complementary base pairs. How are their shapes different? RNA forms complementary base pairs within its strand and forms unique shapes. DNA is a double helix. How do they differ in stability? DNA is more stable than RNA due to its double sugar phosphate backbone. RNA has only one sugar phosphate backbone. How are their locations in the cell different? DNA remains in the nucleus, while the different versions of RNA move through the nucleus and the cytoplasm. Analyze this molecule. How many nucleotides are in the molecule? How many phosphodiester bonds does it have? How many water molecules were released when the molecule was formed? What is the general name for this molecule? Which part of the molecule is the backbone and which part carries information? There are 4 nucleotides identified by the C for cytosine, G for guanine, A for adenine, and U for uracil. How many phosphodiester bonds does it have? 3 bonds identified in yellow. They join the phosphate to the pentose group. A water molecule is released for each bond between the nucleotides. There are 3 bonds for the 4 nucleotides. What is the general name for this molecule? There are 4 nucleotides, so it is a tetranucleotide. Which part of the molecule is the backbone and which part carries information? The backbone is composed of the sugars and the phosphate groups. The nucleotide base sequence carries the information.